Hello everyone. Today I'll give a presentation about our study characterizing the cellular diversity of early development of the human hindbrain and spinal cord. The development of the central nervous system, or CNS, is studied along the rostral-caudal, which is the head-to-toe axis, and the dorsoventral, which is the back-to-belly axis. The rostral-caudal axis encodes the position of the neurons from the head to toe, and this is determined by the remarkably conserved time-dependent expressions of Hox genes. The dorsoventral axis specifies the types and the functions of the different neurons, and this is under the control of specific transcription factors such as FGF, TGF-beta, and while we know that the Hox patterning drives the regional specificity along the rostral-caudal axis, we know very little about the early development of brain and spinal cord in human because we cannot easily access the developing human embryonic tissue. This is due to the ethical reason as well as the technical reason, such as timing of the CNS formation and small numbers of the cells, which makes it difficult to isolate and analyze these tissues from this early in development. So to address this challenge, our collaborators, Dr. Randy Ashton and the postdoc from his group, Dr. Nisha Iyer, have developed a novel in vitro platform to mimic very early human nervous system development. So they have performed a chemically induced differentiation of human neuronal pluripotent stem cell, or HPSC, to the mature neuron cells by a progenitor such as the neuromesodermal progenitor, NMP, or dorsoventral progenitors, and have isolated cell populations at multiple time points, which is from 24 hours to the 216 hours, that correspond to the different positions along the rostral-caudal axis. To characterize these cell populations, we performed a single-cell RNA sequencing experiment to define the cell types on the basis of the transcriptome state. So our dataset comprises around 48,000 of the cells and 24,000 of the genes, spanning the 14 different time points of the rostral-caudal and dorsoventral axis. So with this dataset in hand, we have two overarching questions. So the first, what are the cell types in the human hindbrain and spinal cord? And second, what governs these cell types at the gene expression level? So to answer these questions, we developed a data-driven approach that does not rely heavily on the conventional marker gene-based method. So to address the first question, what is the neuronal cell type diversity? We developed a computational pipeline which consists of two steps of the clustering based on the matrix factorization. So for the first stage, we applied the matrix factorization to all the cells in our dataset to identify the major patterns that we think correspond to the cardinal cell types. And then in the second stage, we reclustered different subset of cluster found from the first step to find the final subpopulations. And finally, to identify sets of the genes that are associated with these different cell types, we applied an approach based on the multitask clustering that can identify groups of the genes associated with the sets of the cell types. So our clustering approach is based on the non-negative matrix factorization, or NMF. So NMF is a popular data analysis technique, especially suitable for the sparse and high-dimensional non-negative data set, such like the single-cell RNA-seq count matrices. In NMF, our objective minimizes the difference between the original matrix X and its approximation from taking the product of two lower dimensional non-negative matrices U and V. And U and V have a much lower dimension than comparing to the original matrix X, and they encode the major pattern in the matrix X, which they can also be used for the clustering. So in particular, after we apply the NMF to our single-cell RNA-seq data matrix, our two lower dimensional matrices U and V are representations of the cells and genes respectively. And each of the columns of the U matrix capture the latent transcriptome signature, which are related to the cell type specificities. So therefore, by examining these matrices, you can get out the cell cluster. And furthermore, typically this gives a good initialization for the k-means style algorithm. So cleaner clusters can be obtained by applying the k-means to the lower dimensional matrix. So we apply the NMF-based clustering on our entire dataset. So what you see here is a t-SNP plot where each dot is a cell and each color represents one of the 25 different clusters. So we determined the 25 to be the proper number of the clusters by scanning a range of the k-values and examining the clusters using the different cluster evaluation criteria and the numbers of the distinct patterns of the expression. So we first tested the efficiency of our induction protocol of neuronal population by identifying the cells expressing the general neuronal marker gene SNAP25. And as you can see here, we see that the majority of our cells are mature neurons, which are expressing the SNAP25 genes. Next, to relate these cell clusters to the cardinal cell types, we examine the sample compositions of each cell cluster. We find that our clusters capture the major source of the variation along the rostral caudal and dorsal ventral axis, and typically overlap clusters from a few neighboring time points. So this implies that our samples and our clusters recapitulate major neuronal cell types that emerge along the developmental time points of the rostral caudal and dorsal ventral axis. Next, we examine the expression of Hox genes, which determines the time-dependent region-level specification along the rostral caudal axis. So for example, as you can see here, we see the Hox A2, D4, A5, which is depicted in the top part of the bubble plot, are associated with the hindbrain, whereas the C6, C8, C9 are associated with the cervical or thoracic spinal cord. And the clusters expresses the different combination of Hox genes that are known to be associated with the different regions from the hindbrain to the spinal cord. And finally, we check the expression of known marker genes of specific cardinal cell types in our clusters. So we can see several of our clusters express localized expression of key marker genes, such as LHX9, which marks for the DL1 cell type, is expressed in the clusters 7 and 10. VSX2, SOX14 for the V2A cell type are expressed in the clusters 12, 14, 22. And the MNX1 for the motor neuron is expressed in the clusters 16, 17, and 20. So overall, based on the marker gene expression we find, each cluster comprises a high proportion of one or two known marker gene expressing cells. However, we also observed that there are still large numbers of unknown cells in the clusters, which are shown as a gray in the bar plot. And also there are several cell types, such as motor neurons shown in the green here, are shared by multiple cardinal cell clusters. So this suggests that there are likely additional finite populations of each of these identified cell types. So therefore, we carried out another round of the clustering based on the consensus clustering to identify the finite clusters associated with the subpopulation in our first round of the clusters. So the first step of the second round of clustering was extracting the specific cell clusters based on their cell type annotation. So for example, there are 5 clusters that represent motor neurons cells in our dataset. So we extracted these clusters from the entire dataset and clustered these further. Because we have smaller numbers of the cells for each cluster, the resulting cluster might not be very stable. So therefore, we developed a consensus clustering approach to define the robust cell clusters. Briefly, we applied multiple rounds of the clustering by changing the algorithms, including the NMF or graph clustering, as well as the changing the parameters, such as the number of the clusters, to obtain the multiple clustering solutions. And then we generated a consensus matrix of cell-cell co-clustering, where each values of this matrix is the proportion of times two cells are in the same cluster. And once we have this symmetric consensus matrix, we apply the NMF clustering once again to find the best clustering structure. So here, again, we apply the range of K values and pick the best based on the evaluation metrics such as stability or silhouette index and the tendency of the clustered consensus matrix to exhibit a block diagonal structure. I'm going to depict this approach with the motor neuron example. So as you can see here, we identify the 7 clusters, and we can clearly see the block diagonal structure upon the consensus matrix. To characterize our final clusters found here, we examined their relationship to each other by doing a hierarchical clustering between the pseudo-bulk gene expression profiles of each cell groups. So we can see a representation of caudal, rostral, and cervical thoracic cell types that are organized in a hierarchical manner. However, to really understand these cell clusters, we need to identify sets of genes for each of these clusters. So this brings me to the last step of our approach, which aims to address the question of which genes or gene set drive different cell types. One way to identify genes associated with the particular cell cluster or cell type is to doing the differential expression analysis in this cluster versus the other clusters. However, this pairwise approach is not scalable when we have a large number of cell clusters that are related. So what we need is a way to consider the genes combinations across the multiple cell types at a time. So then how we do this? Here we simply repurpose our previously developed multitask clustering framework algorithm, which was developed to identify the gene modules on a hierarchy such as phylogeny. So in our setting, we use the hierarchical relationship among the cell clusters while simultaneously grouping the genes into a finite numbers of the expression state based on their pseudo-bulk expression in each cell cluster. So once we have defined these gene expression states for all cell clusters, we can identify genes with interesting patterns of expressions on a hierarchy. So for example, for the genes CPNE4 here, we see it is associated with a high expression state in a cluster C7 and C2, which are associated with the rostral head region compared to the other clusters that are corresponding to the lower part of the body. And we can further confirm this by looking at the pseudo-bulk expression as well as the single-cell expression of this gene. So once we can have these expression state patterns, we can group genes based on the similarity of their patterns to identify the different gene sets. And these gene sets can be used for the further study. So for example, for the motor neuron example here, I'm showing you the 2 gene sets, which is the one which is showing that the gradient of the expression along the rostral caudal axis, and the other one is showing the genes upregulated in the caudal thoracic motor neuron specifically. And interestingly, a number of these genes have a regulatory function, such as the transcription factor or signaling proteins. And so they could be the good candidates for the driver genes, which can be further functionally validated. So to summarize what I presented today, we generated a new single-cell RNA-seq dataset of early human hindbrain and spinal cord development. And we developed a computational approach to examine the cellular diversity of a single-cell RNA-seq dataset and candidate genes associated with the different cell clusters. The application of our pipeline to the single-cell RNA-seq dataset defines major cell types along both the rostral caudal and dorsal axis, and identifies gene sets exhibiting the combinatorial patterns of expression across the multiple related cell types. So I'd like to thank to our ROI group members and also our collaborators in this project who generated this fascinating novel dataset, and also our funding resources, especially the James McDonald Foundation and the UW Data Science Foundation. Thank you for the paying attention, and we'll be happy to have the questions.